Hey Bio30s, and welcome to our sixth and final video on the classical genetics unit. Today I'm going to be discussing mainly a concept known as pedigrees and how we can use pedigrees to calculate modes of transmission depending on whether we have autosomal recessive, autosomal dominant, or X-linked recessive or X-linked dominant transmission of a trait based on the conversations we've already had earlier in the unit. Before I get started though, last video I should have told you to watch a quick review video. I'll get you to watch that video right now. It's a crash course video on heredity, which is going to basically recap every concept that was sort of talked about in the first 5 videos of the unit. So if you feel like you need a refresher, great video to watch before we actually move on today. And before I get started on the topic of pedigrees, I'd like to quickly just recap a little bit about our look back on our molecular genetics unit. We did discuss genetic engineering, being able to use recombinant DNA to basically splice DNA from different organisms together to get GMOs. Before there was that, and I discussed this a little bit earlier, we did discuss briefly, and I discussed it last year in Bio 20, something called artificial selection or artificial breeding, which has been going on for thousands of years. Humans have been basically been breeding for selective traits in a lot of agricultural products or agricultural strains of plants and of animals. The example in the notes here talks about corn. You can see the earliest versions of wild corn here that did not have a lot of kernels on the head. Humans over time began to breed to maximize the yield on the head of corn until we see the corn that we recognize and know today. But it looks very different from the wild strains that were basically taken by humans initially, and when they began cultivating them in Mesoamerica over thousands of years. So it took a while, but again, this is a more natural way to, even though it's called artificial selection, we're not basically snipping and piecing pieces of DNA together. We can create new strains of an organism by simply breeding for selective traits. I mean, dogs would be an example of that, breeds of dogs, breeds of cats. And this isn't something that's an ancient concept. Recently, farmers in Saskatchewan use artificial selection to create a strain or a version of rapeseed that was too toxic for consumption. Well, it turned out to be a gold mine. Even though you couldn't consume it officially, the oil that it produced was one of the healthiest types of oils known on the planet. And you know this plant, it's canola. So canola, which grows all over Alberta and Saskatchewan, it was developed through a process of artificial selection. And farmers in Canada are still using artificial selectional techniques to create leaner meat in cattle, to create leaner meat in hog or pork products. So this is something that's still being used today, even though it's sort of the ancient version of genetic engineering. It's still being used in great, in a lot of ways, even today in the 21st century. What I do want to discuss though today, for the rest of the lesson today, is a concept known as what is called a pedigree. Now pedigrees are essentially fancy family trees that allow us to screen and detect the possibility if there is a genetic disorder that runs in a family. Maybe a gene is being carried down from generation to generation, and that's causing members of the family to basically be afflicted with some kind of a genetic disorder. Pedigrees, once you know the individuals in a family, you can map out which individuals have a disorder, or you can maybe figure out that a person in the family is a carrier of a disorder. And based on the pattern of which individuals in the family get the disease and which ones don't, we can kind of determine which gene and where the gene is that's causing the disorder. Is it a gene that's found on one of the chromosome pairs 1 to 22, an autosomal disorder? Is it dominant? Is it a recessive disorder? Or is it a dominant or recessive disorder that is located within, let's say, one of the the X chromosomes on chromosome 23. For this course, we're not going to be discussing Y-linked disorders, but be aware that if there was a Y-linked disorder, we would also be able to figure out patterns that would develop. For example, in a Y-linked disorder, you would only see males being affected. There would be no females at all. There wouldn't be female carriers. Only, essentially, if a male in the family has it, every male in that family would get the disorder because it would be passed on from the father to the son through the Y chromosome. So that's one that we're not going to be discussing, but we will be discussing the patterns of autosomal recessive, autosomal dominant, X-linked recessive, and X-linked dominant here today. But before we do that, let's take a look at a pedigree and just how do we kind of navigate one. On this slide here are a bunch of the symbols that are used on pedigrees, and don't worry, a lot of these symbols are going to be located on your Bio30 data sheet. So if you've got your bio data sheet kicking around, or go onto my website and link to the Bio30 data sheet, you can see what symbols you're given as to what you don't have to memorize. But there's certain things that you should be able to do without looking at your data sheet. For example, males are going to be squares in the pedigree. Females are going to be circular. Horizontal lines are going to indicate if they're between two symbols like this. Not this wouldn't count, but this line here between the actual shapes, that's going to indicate a mating. The line heading down would then indicate that these are the offspring of these two individuals. So these would be the individuals in generation 1, indicated by a Roman numeral 1. These would be the individuals in generation 2, indicated by a Roman numeral 2. And this is the individual in generation 3, indicated by a Roman numeral 3. We can also identify each of the individuals on each generation from left to right. We can give them regular numbers. So if I have to identify someone in the pedigree, I basically number them from left to right. So this person would be 1 1 1 2 1 1 1 2, sorry, 2 1 2 2 2 3 2 4 2 5, and this would be 3 1. So when you're identifying people, now when you see a pedigree, the numbers aren't necessarily going to be there, but you just know that you're counting one. You're counting in the order from left to right on each of the generations to identify an individual. So if I were to ask you to find individual 2 5, first generation, second generation, 1 2 3 4 5. So that would be 2 5. That's how I would find someone in the pedigree. Let's look at some general things. Parents that are in generation 1, how many kids do they have? Think about it. Pause me if you need to. How many kids do parents in generation 1 have? They've got 4. Even though there are 5 individuals on this generation, you can see that one of them is not an offspring of these parents. It is a spouse or a mating partner of one of the children. So this person here is not one of the children of the parents in generation 1. How many sons and daughters do they have? In generation 1, how many sons and daughters do they have? Pause me if you need to. There are going to be 2 daughters, and there are going to be 2 sons. 2 daughters, 2 sons. Trick question, this one you may not know. Which of these children, can we tell which children is the youngest and which one is the youngest? Can we tell that by looking at the pedigree? In theory, yes, we can. Oldest children are always going to be on the left. Youngest children are always going to be on the right. So the children are listed in the order of their birth. That's how you know which of the children is the firstborn, and which would be, in this case, the second, third, and fourth born. Even though the numbers 3, 4, and 5, technically it's the first, second, third, and fourth born. But they're still identified by the numbers from left to right. What's messing this up here is there was a spouse on that generation that's causing the numbers to be off by 1. And there's other symbols there that you can see. If you see a symbol like this on a pedigree, if it's like a black circle or a black square, that's going to indicate that the person is suffering from a disorder. They actually have the phenotype of the disorder, whether it's a recessive or dominant disorder, doesn't matter. If the person, let's say, has hemophilia, which is X-linked recessive, that person on a pedigree, and that would indicate that person has hemophilia. If they were so kind as to do this, if they gave you this coloration, and they don't always do this, because sometimes you have to figure that out for yourself based on the pedigree. If you were to see a half and half coloration, that indicates that the person is in fact a carrier of the disorder. Now, I've chosen a very bad symbol here. This person would be, let's say, a male carrier of a autosomal recessive allele. So this person would be essentially heterozygous, one dominant, one recessive allele. Although they're not always going to show you that in the pedigree. Okay, let's take a look at the pedigree that I've drawn and let's see if we can figure some stuff out. I believe it was not, was it last day? I don't remember if it was the last lesson or not. I cannot remember, but it was one of the last couple of lessons I discussed blood typing. I remember. It was the video on codominance and incomplete dominance. So if you haven't seen that video, you can watch that video right here about complete and incomplete dominance. I discussed blood typing is one of the last things in that video. And we discussed how you calculated a person's blood type. Well, here I've got a pedigree that's going to basically show us, blood typing. So I'm going to get rid of these squares here in this circle here just so I can kind of get rid of it. What I want you to do is based on the pedigree, I've given you blood types. I got a person who's blood type A, blood type B, blood type B, blood type O, blood type B, blood type O. Based on that, I would like you to try to calculate all the genotypes of all the individuals in this pedigree, and including the parents, which I have not given you a blood type for. So I'd like you to, when you're ready, pause the video, and then I want you to see if you can calculate all the genotypes of the individuals in this pedigree. Ready, set, go, and here comes the answer. Okay, so are there a couple people that I know the genotype of right away? Well, when I'm doing blood typing, blood type O is always my friend. I always know the person who's a blood type O is going to be little I, little I. This person is going to be little I, little I. So I've got two people that I know the genotypes of for sure. Because of that, I now have information about other people in the pedigree. For example, this person here is little I, little I. Well, that means that that person had to have gotten at least one little I from each of the parents. Excuse me. So I know that this person is little I and that person has a little I. Now, because this person has blood type B, I automatically know what the other allele is. It's going to be a capital IB, and this person is blood type A. They're going to be capital IA. So there's those people. Now I know the genotypes of. I'm down to now 4 people that I need to know the genotypes of right now. I know that I've got a blood type B, so that means that this person is going to be IB for sure, at least one allele. And these people are going to be IB. That's perfect. And now the rest, I don't know everything. Here's what I do know. These parents have a child that has an O blood type. O blood type. This means that these parents have at least one little I each. That I do know. As for the rest, I don't know. I know that because they have children that are IB and also IA in their genotypes, I know for a fact that one of the parents has to be A and one of the parents has to be B. This person got this IA from one of these two parents, and these people got the IB from the other parent. Neither of these parents can be AB, because if that were the case, they couldn't have had a child who was O. So this person is either going to be IAI, or and this person is going to be IAB, or those genotypes could be switched. At this point, I honestly don't know which parent has which genotype, but I do know that those are the two genotypes that are going to be found in that generation. I would need to know more information about their family trees to make a better determination of whether or not they were if this parent was A or this parent was B. But I know for a fact that at least one of those are the genotypes. I just don't know on which side those genotypes are falling. Which leaves me now with this individual and this individual. I didn't know if, and you see that I only put down IB and IB. I didn't know if they were IBIB or IB little I. Well, I now know that. Because the parents up here there's only one IB allele between the two parents, I know that these guys down here, or this guy, pardon me, and this gal, they have to be heterozygous. So just by looking at the pedigree and knowing the phenotypes that were in the pedigree, I was able to use that pedigree to help me determine the blood types of pretty much everybody in the family, with the exception of the parents. I don't know if it's the mother or the father who has either A or B. They have those blood types. We just don't know which parent has which at this point. That's something that's the only part of this pedigree that is uncertain at this point. But all the other genotypes now are basically locked in. We know those genotypes, and that was just by using the pedigree, as I just mentioned. All right. So when we're using pedigrees, we can also look at pedigrees and we can see patterns. We can see patterns in the pedigrees, and this is where I would encourage you to make sure you've got your notes open, because these pedigrees, I don't have them written on the board, but they are on the slides, and the slides will definitely help you kind of see what I'm talking about. The first type of transmission that we'll be discussing is autosomal recessive, and that goes all the way back to our first discussion, Mendelian genetics, complete dominance. Either in order for a person to have a recessive disorder, they're going to have 2 copies of a recessive allele. They have to be homozygous recessive. That's the only way that a person in this family or in the pedigree would have the condition. I know, for example, in this pedigree down here, and I'm just going to read off the numbers off my screen, I got 1, 2, 1, 3, and 3, 2 that I know on that diagram are all basically going to be homozygous recessive, because, excuse me, they are all colored in. I know that they are affected individuals. The second generation, you'll notice, has a dot in the middle of all of those. So 2, 1, 2, 2, 2, 3, 2, 4, 2, 5, all have black dots inside of their respective squares or circles. That's another way to show that they're carriers. So the second generation are all heterozygous carriers of the disorder. You can see that because of it, and this is, Gregor Mendel saw this with his peas, certain traits would skip a generation. If it was a recessive trait, it would skip a generation. We're going to definitely see skipping of generations with autosomal recessiveness. But you can see that both males and females are being affected. We have an equal number of males and females being affected, and both males and females can be carriers of the disorder. You can certainly be a carrier for this disorder if you're a male or female, because if you have only 1 copy of the allele, you're now a carrier, and that's why it's going to skip potentially to the next generation. You have 2 carriers that are heterozygous. You got a 25% chance, a 1 in 4 chance of the offspring are going to be homozygous recessive if you run the Punnett square for that. So when you see autosomal recessive in a pedigree, you're looking for skipping of generations, and you're looking for the fact that both males and females are being affected relatively the same. And can this trait be passed down from a mother to a son? Not necessarily, but you would need at least 2 carriers in the parents. Both the parents have to be carriers, or both the parents have to be affected, or one's affected and one's a carrier, in order for their offspring to have the disorder. The key here is the offspring, whoever has the disease, has to have 2 copies of the allele. Otherwise, you're not going to get the disease. So that's something to keep in mind there. An example of this one, by the way, is cystic fibrosis. That's a great example of an autosomal recessive inherited disease. Well, let's take a look now at autosomal dominant. Is the gene found? Again, autosomal refers to any disorder that's found on chromosomes pairs 1 to 22. It's those first 22 chromosomes where you're going to find these genes. If it's autosomal dominant, each affected individual has to have at least 1 affected parent. If I have a person who has the disorder and neither of the parents has the disease, it can't be autosomal dominant. So that's something you're looking for in this, is the fact that a person who's affected has to have at least 1 parent who has the disorder. If you don't see that, then it's definitely not going to be autosomal dominant. Generally speaking, the disorder does not skip generations. It can, but not. It's not as likely to skip generations as it would be if it was autosomal recessive. Is much more likely to skip generations than autosomal dominant, and you're going to see, just like in autosomal recessive disorders, equal numbers of males and females are going to be affected. You're going to see both genders being affected relatively equally. An example of this kind of disorder, autosomal dominant, would be diabetes insipidus. We talked about that in the endocrine unit. That is a lack of the production of ADH coming from the hypothalamus, which is being released by the posterior pituitary. That's diabetes insipidus. You have very large amounts of urine production because you're not producing ADH properly. So that would be an example of such a disorder. All right. What about X-linked recessive? What about our X-linked disorders? Well, here's where the gender splits with the people who are affected is not going to be equal. If you see a pedigree and males and females look like there's a skew there, where maybe more males are being affected than females or vice versa, at that point you want to maybe seriously consider looking at the possibility that that pedigree is showing some kind of an X-linked disorder, whether it's recessive or dominant. I'll explain the differences between those in a second. X-linked recessive, and a great example of X-linked recessive, which I've talked about already in this unit, is hemophilia. I've talked about hemophilia back in Bio 20. It's where you don't clot your blood properly. Women are carriers. Men cannot be carriers of X-linked recessive disorders. If a male has a single copy of the X-linked recessive allele, they've got the disease. A male only requires one allele. Even though it's recessive, they only require a single allele. A female will require two recessive alleles to basically get the disorder. And I talked about that last day. I talked about that in the X-linked and multiple allele video that I showed. That was the last video you would have watched. We talked about that males are more likely to get X-linked recessive disorders than females, because females require both of their parents have to have the allele in order to have a female who gets the disorder. So the mother would either have to be a hemophiliac in this case, or a hemophiliac carrier, and the father would definitely have to be hemophiliac in order for a daughter to get hemophilia. So that's something that you got to keep in mind. The mothers can pass the condition down to their sons. So if I have a mother who's a carrier, she's not a hemophiliac, but if she's a carrier, there's a 50% chance that she's going to pass that gene down to the son, because she's got a 1 in 2 shot of passing that hemophilia X chromosome down to the son. More males, again, are affected by females. Affected males, here's an important one. If the father is affected with hemophilia, because he's got the single copy of the X chromosome, he cannot pass that down to a son. So a father cannot pass the disease down to his son, but a father can pass the disease potentially to his daughter, but again, only if the mother is also a carrier of the gene, or if the mother is hemophiliac as well. So the father alone cannot pass the disorder down to his daughter. That can't happen in an X-linked recessive inheritance pattern. Can you see skipping of generations? Yeah, just like autosomal recessive. So, but you have to look at the potential here of the skewing of the affected individuals, right? Who can be a carrier? Only females can be carriers. Males cannot be carriers of these disorders. So that's something to keep in mind. And the fact that males are way more likely to get affected by these disorders as opposed to females. An example of this, and it's the classic example that's always talked about, Queen Victoria. Queen Victoria was basically a carrier of the hemophilia gene, and she passed down, she had 5 daughters, 2 of which she gave the allele to. So 2 of her 5 daughters were carriers of hemophilia, and then they passed that mutation down to various houses across the continent. A lot of families were affected heavily. One of them in particular, and I'm just making sure they're on the list here. Yes, here we go. They passed it on to the Romanov dynasty in Russia. The last Tsar of Russia, Nicholas II, before he was killed by the Bolsheviks in 1917, his son was a severe hemophiliac who may not have survived to adulthood because he was, had a real issue with hemophilia. But that hemophilia gene that was in the family of the Romanovs, the Tsars of Russia, that hemophilia gene came from the house of Queen Victoria. It was her family who brought that gene into the Romanov dynasty, and then the males became affected by the hemophilia gene in the Romanovs, not the females, because again, you require 2 copies of the allele for the females to be affected, because it was a X-linked recessive disorder. And the final one is X-linked dominant, which is very rare. An example of this would be vitamin D resistant rickets, which is an example of this disorder. Generally speaking, unlike X-linked recessive, more women are affected by X-linked dominant than men. So more females tend to be affected by X-linked dominant than males. So there's a skewing in that result towards the side of the females. All daughters that are affected, all daughters of affected males are going to be affected as well. So if remember, just like in X-linked recessive, if I'm a male, I only require 1 copy of the allele to basically become affected by the disorder. But unlike X-linked recessive, females also only require 1 copy of the allele to become affected. So there are no carriers in this disorder because it's dominant. Anytime you have a dominant disorder, there are no carriers. You either have the disorder or you don't have the allele. There is no middle ground with these disorders. So again, all daughters of affected males are going to be affected as well. Fathers cannot pass the condition down to their sons. Only the mother can. So again, just like in X-linked recessive, males cannot give males in their family the disorder. So if I have a female that is not affected and a male that's affected, and I see a pedigree where a male in the next generation is affected, it can't be X-linked dominant because a male who's affected can't give the allele to his son because he's going to give the Y allele to his son, not the X one. He's going to get the X chromosome from his mother, and if the mother doesn't have the disorder, there's a 0% chance that she's carrying it because there are no carriers. So that's another way you can kind of see. So using these pedigrees, the kind of question you're going to get, this is what this is all sort of leading up to, is you're going to get a question, presumably at some point in a unit exam or on a final or on a diploma exam, that's going to have it's going to show you a pedigree. It's going to say, here are 4 pedigrees. Which of these is an example of an X-linked recessive? Which of these pedigrees is an example of an autosomal recessive trait? And you have to be able to kind of figure it out by looking at, which individuals are affected, who could be carriers. Sort of what we did with the pedigree at the very beginning today, except we use blood types today. But we sort of break apart the pedigree and figure out who on the pedigree is carrying which alleles. And in the case of today, we figured out the alleles for blood typing, but it might be a question about, here's a pedigree showing hemophilia, which is X-linked recessive. Which of these pedigrees is going to be an example of a family that's suffering from hemophilia? And then based on this information, you have to kind of figure out which of the pedigrees is going to be the one that fits the bill. So that's basically that's basically it. That's the unit. We're finished. We've got one more booklet coming your way before the review, before the end here. Today, based on today's conversation, can you complete for me the booklet entitled Pedigrees? It's going to have quite a few pedigrees for you to work on, put together, work through, see how well you do with those. And that's pretty much it for today. That's it for the unit on classical genetics. The next time we'll check in, I'll be starting on the 7th and final unit in Bio 30. We'll be starting our look at population genetics. Until then, thanks for tuning in, and we will see you soon.